All right, in this video, we'll continue the topic of genetics, moving on to the human genome. So first, we'll look at just human chromosomes, a couple of facts concerning human chromosomes. There are 46 chromosomes in most human cells, 23 homologous pairs. We talked about this idea of homologous pairs already. 22 of those homologous pairs are called autosomes. These are just regular everyday chromosomes, and then one pair of those is called sex chromosomes, and these can be either XX for female or XY for male. A picture of chromosomes is called a karyotype, and here you see a karyotype, and here you see another karyotype. This particular karyotype is one of a male. You can see here a single X chromosome, and a Y chromosome is this little guy over here. But otherwise, there are 22 homologous pairs, and these are your autosomes. Here is a female karyotype, and you see the two X chromosomes and no Y. And notice here you have 3 on the 21st pair. We'll talk a little bit about what that's happening, or what's going on here in just a bit. And so this just shows you sex chromosomes. This is something that we've already looked at, and how sexual traits are how sexual traits are determined. But this is just also to show you that when sperm and egg combined, there's a 50% chance that the individual will be female and a 50% chance that the individual will be male. Human traits, most human traits are autosomal traits, meaning that they are inherited on autosomes, but there are some traits that are inherited on sex chromosomes. Most traits are just coded for on these chromosomes, like these that you see here, widow's peak, hanging earlobes, some of these other things that I've never heard of, like Darwin's point, but you can see if you have those things. And then some traits are kind of both. Some traits are not inherited, or maybe the part of it is inherited, but they can also be attributed to the environment, nutrition, the presence of mutations. Size is one that is important here. Some people are born with big, as far as their genes lead them to be a bigger person, but how they carry their weight is going to be a product of their nutrition and their environment and so forth. A little bit about blood types with humans. We have 4 blood types. They're A, B, AB, and O. A and B are codominant. Thus, when they're inherited together, they're both fully expressed. And so if mom gives you A and dad gives you B, then you will be AB. There's not type C, there's not a blending, and so both are fully expressed. And then A and B are both dominant. O is recessive, and so you can be a carrier for type O. You can be, say, type A, but have an O gene. So you can have type AO blood, even though you wouldn't be called that, but you could pass the O down. And this just shows you, and we're not going into this really deeply, but just shows you if you have type A blood, you have anti-B antigens. If you have type B blood, you have anti-A antigens. And this means that you can't have A or B. If you have A, you can't have B, and vice versa. If you have AB, you obviously have both of those types, so you have none of those antigens. And so if you have AB, you can actually accept any kind of blood. Well, if you have type O blood, your blood isn't recognized as A or B, and so it's universally accepted. You're considered the universal donor if you are type O, but you have antibodies for A and B, so you can't accept anything but O. So that's just an interesting fact there. Another factor with blood is called the Rh factor, or the rhesus factor, which is named for the rhesus monkey, which is what you see there. And a lot of times you'll hear someone is type O negative or A positive. Well, the Rh factor is another factor in blood typing, and the positive is a dominant trait and negative is a recessive trait. And so if you have the trait, you are either homozygous dominant for it, big R, or you're heterozygous for it, meaning that you're positive yourself, but you could possibly pass down negative to a child. And so you could have children with the negative trait. And this brings us to pedigrees. A pedigree is just a chart that allows traits to be tracked from one generation to the next. Typically, you see it like this, whereas the male is a square and the female is a circle, and the shaded shapes have to do with the trait that's being tracked in this particular pedigree. There is something called hypercholesterolemia, and it is an autosomal dominant disease. You can read those things and you can see how it would be tracked. Dad has it, our grandpa has it up there at the top, pass it to his son, daughter, and other daughter, but a couple of his kids don't have it because mom didn't have it. You can kind of figure out what their genotypes would be just by solving a little puzzle there. And you can also do it for recessive traits as well. Notice here, mom and dad don't have the disorder, but little Bobby does because, well, mom and dad both were carriers, and so little Bobby got it. And you can see here, this kid doesn't have it, but his wife did, and so one of their kids did. And so you can kind of track through that. Couple of disorders that are associated with genetics. We typically call these genetic disorders. Surprise, surprise. And these are just things that are codiform of genes. They're not like a disease that you get that can be cured, more so something that all of your cells have in them. So you can only deal with the symptoms. You can't actually cure the disease. And so there are some that are called recessive disorders because they're carried by recessive alleles. You can see some of the examples there. We're not going to go into detail on these. Tay-Sachs is one that affects infants. Cystic fibrosis actually this can drastically affect life expectancy. And then there's dominant disorders as well. Achondroplasia is just a form of dwarfism, but it's a dominant disorder. And then Huntington's is a genetic or is a nervous system disorder. There are some that are codominant as well. Sickle cell anemia has some codominant factors in it, and it's a really big thing. But there are some codominant factors associated with sickle cell anemia, but not as big of a deal as these dominant recessive disorders. And then I think I have some pictures of them. ALS is one that is a chromosomal disorder. It's one that's affected on the 21st chromosome. The average, let me see if I got that. No, I don't. Okay, never mind. So 21st chromosome, really small chromosome, but it causes a lot of health problems, and ALS is one of them. And ALS is commonly called Lou Gehrig's disease, which is why I have a picture of Lou Gehrig here. And let's talk a little bit about sex-linked genes. We talked some about this maybe in the Punnett Square video. And basically, these are genes that are only found on the X or Y chromosome. So if you go back here to the very front, and all these are autosomal genes, and down here you have the X chromosome. Well, the X chromosome is necessary for life. Notice males and females both have it. There's no such thing as a person who's living and breathing today that doesn't have an X chromosome. There are a lot of people that don't have Y chromosomes. They're called females. But the X chromosome is something that is necessary for life. And so a lot of things that we talk about can be called sex-linked genes, in that they're genes that are located on one of these chromosomes, but there are also sex-linked disorders because it is coded for typically on the X chromosome. And so when someone has that, it can drastically affect them. Color blindness is one example. Hemophilia is another one. And another thing about X chromosomes is because females have two, one of them is kind of extra, and so they will actually inactivate one of their X chromosomes, and it will become something called a bar body in their nuclei. And so it will just kind of be of a turned off X chromosome. One interesting thing about the Barr body is this is what causes calico cats, which is why you don't have any male calico cats, or that's really rare. Another chromosomal disorder is called nondisjunction. Big easy definition for nondisjunction is when chromosomes fail to separate during meiosis. All right, it can happen during meiosis 1, and that's what you see here, where homologous pairs separate and they didn't separate. See, dark blue and light blue should have separated here, but they both went the same way. And so you have a couple of cells that have extra chromosomes and a couple of cells that have too few chromosomes. And so this can lead to problems like what we saw back on here. So this particular individual had too many 21st chromosomes in one of the gametes. So the egg maybe had 2, the sperm cell had 1, they combine together, and whereas everything else has 2, the 21st has 3, and we call this trait Down syndrome, or trisomy 3 chromosomes 21. There are a couple of others. If you have non-disjunction occurring on the sex chromosomes, you can see them there. Basically, the idea here is that non-disjunction is a failure for chromosomes to separate either during meiosis 1 or meiosis 2. Big idea. A couple more things. One thing that is growing and growing even more so since I first wrote these notes and did this PowerPoint is this idea of DNA analysis, and this allows for parents to test for the likelihood of childhood disease, and allows for a lot of different things, and even more starting to allow parents to see traits of a child in utero. And so there's some ethical concerns here, but in general, I think you get the idea. There's a lot that can be said and learned from these chromosomes just simply by looking at them and understanding what's going on. And that brings me to the last point here is about the Human Genome Project. In 2003, the Human Genome Project was completed, which seems like forever ago now. But basically, they mapped out the entire human genome and discovered what genes are located and where they are located, and discovered that we have approximately 24,500 genes, which create about 50,000 proteins. Now, there's a lot more proteins that can be made because of some different combinations of things, but that's a lot going on. And what they discovered when they coded for the human genome, and they said, okay, they thought they were going to discover all the answers to the universe, but really they figured out that there's a whole lot more that they don't know. And so there's still a whole lot of study that goes on in this field just to understand the complexities of all the different things that are going on in our chromosomes.